Hello, hi, a very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So here we are on part 3 of biotechnology and its applications. So we are talking about how biotechnology is being used in our practical life. So we have already seen how it is being used in the field of agriculture. So today we'll be talking about how it will be implemented in the field of medicine. So we're going to talk from 3 perspectives: how your insulin is being made, how gene therapy is being used, and how diagnostics can be done. So these are all the topics for today. So we'll try to cover how your biotechnology is used in the field of medicine. Hi W, yes, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. I hope you're doing well, and I also hope you are safe. You are away from all this COVID, because your COVID, again, the lockdown is taking a toll, so the cases are rising. So I hope your place is pretty safe, and please stay safe and take care of your health, because once your health is gone, so nothing is going to help you, right? So please stay safe and boost up your immunity, eat healthy food, right? Yes, so let's talk about biotechnology and its applications. But I also hope you guys remember what we spoke about biotechnology and its application of biotechnology in agriculture. So giving you a quick recap, we only spoke about 2 topics in agriculture. So one was your Bt cotton, the other was your RNA interference, right? So I'll just give you a quick recap of these 2, and then we'll go into the field of medicine. So just try to recollect all of these, okay? So we were talking about how biotechnology is used in the field of agriculture. So it is used in the field of agriculture in 2 ways. One, Bt cotton, to rescue the cotton industry or the cotton plantation from its lower yield from worms, bollworms, cotton worms, etc. The other one is nematodes using your RNA interference. So Bt cotton, so you know India is a very good player in the textile field also. So we have a variety of textiles throughout, and cotton is very basic. So this cotton plants were having very less yield. Yes, you're right, it's Bacillus thuringiensis, absolutely right. But you write B, capital B, and you write small t, so you have changed it properly. It is a typo error. So the genus name should be caps and the species name should be small, okay? Right. So you have a class of insects who feed on the leaves of the cotton plant, like your leptopterans, cleopterans, diplopterans, etc. So now how do I rescue my cotton plant? So thus we go for a particular type of bacteria. So Bt stands for Bacillus thuringiensis. So this bacterium, actually during its life cycle, it produces a protein crystal, which is a protoxin, meaning it is not a complete toxin, it is in the inactive state. Okay, so it is produced inside this insect. Okay. So when the worms eat these bacteria, so this protoxin gets activated. Fine. Hi Shimpi, yes, a very happy good evening. Yes, I am good. How are you doing? So thank you so much for joining my lecture today. So please do like, share and subscribe. I hope Munna is also fine. So say hi to her. Fine. So it gets activated in the gut of the insect. So the gut of the insect has alkaline pH, which activates the toxin. Fine. So this toxin actually creates pores on the gut wall of the insect. So when it creates pores, it causes swelling and leakage of the substance. Okay, so thus it kills the insect and your cotton plant's yield improves. So the gene that codes for this toxin is your 𝐾𝐈 gene. So I can take this 𝐾𝐈 gene and by the process of recombinant DNA technology put it inside the cotton plant so that it can actually be safe from all these worms. Okay. Gut means stomach. GUT is gut and gut means stomach. Okay. So this is how your Bt toxin or your Bt cotton works. So the next thing is your RNA interference. So you know majority in our country also consume tobacco. So when tobacco yield is very less, the tobacco industry is also affected. So mostly the roots of tobacco plants are infected. Okay, so it is infected by a group of worms called as nematodes, especially Maligogna incognita, right? So that is the nematode which infects it. This one. Right? So now how are we going to save the tobacco plant? So we are going to use a technique called as the RNAi. So this is called as the RNA interference. Fine. So this RNA interference means, so this is a natural defense mechanism present in eukaryotes. So what actually happens is, so in the central dogma of life, when your DNA is replicated and the information is copied onto the RNA, we make sure only a single stranded mRNA is there so that it produces protein. So just because the DNA is double-stranded, the mRNA is not double-stranded because a double-stranded RNA will not allow the formation of protein synthesis, right? So we are going to exploit this concept in RNA interference. So whenever my body wants to silence any genes, for example, I'm here, my body has decided, okay, I have to produce this protein. So it is in this process of transcription and then it realizes, oh, I don't need this protein. So my body knows how to silence a gene. My body will produce a similar strand or a complementary strand to this mRNA, thus making it double-stranded mRNA or double-stranded RNA and inhibiting the process of protein synthesis. Okay? Fine. So here what I am going to do. So inside the plant, I take nematode specific genes. So I take the genes such that both the strands of DNA are transcribed. Okay, so in a nutshell, I have double-stranded RNA. Right, so I have double-stranded RNA like this. So there is a particular protein called as the 𝐷ICER protein. So transcribed means the process, the information on DNA is getting copied to the RNA, that is transcribed. So DNA replicates. So this is replication. then you have transcription, and this is translation, right? So the process of copying the information from DNA to the RNA is called as transcription. So now I use a dicer protein to break this down into smaller molecules. So now it becomes small molecules. So these are called as siRNAs, small interfering RNAs. Fine. So now when the nematode comes inside the plant, assume if this is my nematode. So nematode enters inside the plant. According to the central dogma, its DNA will replicate. The information will be copied onto the RNA. So the RNA will be like this. But we have the siRNAs. So the moment your plant senses the nematode entering inside, you have a group of protein called as the RISC complex. So this RISC complex breaks this siRNA into single strands like this. Okay, so now if the nematode produces an RNA like this, we have the complementary pair here. Obviously plus and minus attracts, it directly goes and binds like this, thus inhibiting the protein synthesis. So thus the nematode dies. So RISC is RNA induced gene silencing. C stands for complex. Okay, so this is the entire picture that we read in terms of agriculture. So now how it is going to help in the application or in the field of medicine is what we are going to see. So our DNA technological processes are of immense impact in healthcare, enabling mass production of safe and more therapeutic drugs. Today, everyone is in need of drugs, right? So even if you want to stay healthy, you need drugs. Even if you want to stay or even if you want to cure from a disease, you need drugs. So in olden days, you needed drugs only when you had fever. Today, we have supplement tablets for well-being. Hurry, eat fruits? No. Hurry, eat vegetables? No. No, people prefer or believe the tablets because they feel that can ail you faster. But I don't know how far it is true, but that is how the world is, right? So we go for a lot of supplements. We believe the fruits and the vegetables we eat are not healthy enough. So probably or perhaps, I don't know. So recombinant therapeutics do not induce unwanted immunological responses because they are still safe. Because I am not taking a donkey's protein and giving it to you. I am using the same human protein and I am cloning it in a different way and I am giving it to you. So this is very safe because they do not induce unwanted immunological responses, as in case of similar processes that are not isolated from human sources. So if I take pancreas from a pig and insert it, yes, I have immunological reaction because it is a way different species. It is not of the same species, right? So even if I have an identical twin, if I take the tissue of that identical twin and put it in me, there will be some amount of immune reaction. So to overcome the immune reaction, recombinant therapeutic drugs were a very, very useful one. So at present, you have 30 recombinant therapeutics that is approved for the use worldwide. In India, out of the 30, 12 are being marketed. So you have a lot of recombinant proteins. Let's see one by one. So in medicine, we will see how genetically engineered insulin is produced, how or what is gene therapy. So probably we'll see only these two techniques. Molecular diagnostics is a bit big because we have to talk about PCR, ELISA as well as your hybridization technology. So we will keep that for the next lecture. What is enabling mass? So enabling mass is larger production. Mass production means large production. Enabling is to be able to produce it in large amounts. Okay? Yes. So these are the three ways how it is being used in medicine. So the first one, genetically engineered insulin. So management of adult onset diabetes includes taking insulin at regular time intervals. So you already know India is the diabetes capital of the world, right? So everywhere diabetes is very common in adults, in teenagers, in babies, in infants, in the child that is being born, in pregnant mothers. So diabetes is pretty common. So what comes to the cure of diabetes? So one common cause that we all know why diabetes is happening is because your body is not able to have enough production of insulin, because insulin is a peptide hormone that decreases the blood sugar level, right? So in that case, we need something called as insulin to maintain the blood sugar level. It cannot be pretty high, right? So what would a diabetic patient do if enough human insulin was not available? So would the insulin isolated from some other animal, say for example a cow or a pig or a dog, be just as effective as that secreted by the human body itself? So always original is original, right? Yes. Evolution ho gaya? Yes, I guess I did evolution. I taught Hardy-Weinberg. No, I never taught evolution in New Beginning series, so I will do evolution. I will check once, because I've been teaching for so many classes, I really don't remember. So I think I did only 2 chapters of genetics and I came to biotechnology. So in between I also thought about, okay, I think I have to do evolution, okay, right. So, so original is original, so duplicate is always duplicate, so you cannot change it, right? So would it not elicit an immune response in human body? Okay, iske baad you need evolution. Okay, let me think about it. Sure, W, fine. So what can be the possible solution? Is a bacterium available to make human insulin? Okay, so before we go see how insulin is being made in bacteria, so let's talk about the insulin hormone as such. Evolution is really hard, so evolution is really interesting. Don't worry, I will teach you, okay? Yeah. So insulin used for diabetes was earlier extracted from the pancreas of slaughtered cattle or pig. So slaughtered cattle is when you cut it for your meat or your other purposes, so that is called the slaughtering. So you can take it from the cow or pigs, but this created cross reactions and patients started developing allergy. So you couldn't probably use the insulin from some other species or from a neighboring species. So talking about insulin as a hormone, so insulin has 2 short polypeptide chain. So it has a chain A and it has a chain B, okay, and it is linked together by disulfide bridges. So this is how the structure of insulin is. So you have chain A, insulin is a peptide hormone, so it has a peptide chain A and it has a peptide chain B. Okay, so it has a series of amino acids arranged and the 2 peptide chains are linked by your disulfide bonds. Alright, okay, so here it is. So your insulin that is produced in your body has a pro-insulin state, that is a pre-insulin stage. This is my final insulin, but before the final insulin is formed, it has a pro-insulin stage. So this pro-insulin stage has the chain A and chain B, but it also has an extra stretch. If you can see the grey portion, so this grey portion is called as the C stretch or the free C peptide. But in the mature stage, this extra stretch is cut off and you have chain A and chain B, right? Yes. So, in mammals, including human, insulin is synthesized as a prohormone. The prohormone contains an extra C peptide, but this C stretch is not present in the mature state. So, it is removed during the maturation process inside your body. Now, how am I supposed to make this artificially? It is not possible, right? How can I get this assembled? So probably if I am taking the insulin, so I can take it in this stage or this stage, right? But we are going to use recombinant DNA technology, understand. So if I take the insulin producing gene, it will produce in the pro-insulin form. So in the bacteria, it will be in this form. If I do rDNA technology, we know rDNA technology, I am taking the insulin producing human gene and putting it inside a bacteria. So it will produce the pro-insulin only. It will not produce insulin for me. So now how am I going to chop this off? How will the maturation step happen? Yes, so this gray color is my C stretch or my C peptide, which is removed in my mature stage. Okay, so the challenge is getting insulin assembled into the mature form. Fine. So how did it happen, or who did this? In 1883, Eli Lilly, an American company, prepared two sequences, A and B chains of human insulin. So introduced them in the plasmids of E. coli to produce insulin chains. So there was one company in 1883, that is Eli Lilly. So this company, they took the gene that codes for the peptide A of insulin and peptide B of insulin separately. So the insulin gene is together one, but some portion of that insulin gene codes for peptide A and some portion codes for peptide B. So they took only those portions and they cloned it inside a bacteria. So the bacteria produced peptide A and this bacteria produced peptide B. So now they joined it in lab artificially by disulfide bond. So now your recombinant insulin is ready. They do not have to worry about the C stretch. So chain A and chain B were produced separately, extracted and combined by creating disulfide bond. So this was much easier if you understand. So I have a stretch of gene. So this is my insulin gene. So this insulin gene actually codes for peptide chain A, peptide chain B and peptide chain C. But from my human genome project, I know which part codes for peptide A and which part codes for peptide B. So I isolated only this portion. So this portion which codes for chain A, I cloned it inside a bacterium. I cloned it inside a plasmid and put it inside a bacterium. So B also I have cloned it inside a bacterium. So now this bacterium will produce peptide chain A. So this bacterium will produce peptide chain B. Right? So now I can isolate these two and I can artificially join them with the help of disulfide bonds. So this is how my first recombinant insulin was created. So since it is human insulin, it is also known as humulin. Fine? Right. So first half of it is done. The next one is gene therapy. So we saw how biotechnology is used in the process of production of recombinant insulin or to treat diabetes. So now the next part is gene therapy. So how it is being used. So there are people who are born with hereditary disorders. So hereditary disorders pass from one generation to another generation. With us having studied genetics in the previous lecture, so we know what are hereditary diseases. It passes from one generation to the other, right? And they can have corrective therapy. So could these hereditary diseases have a possible therapy? So attempt to do this is your gene therapy. So gene therapy is a collection of methods that allows correction of a gene defect diagnosed in a child or an embryo. So genes inserted into a person's cell or tissue to treat a disease. For example, if one gene is absent in a person or a child who has a hereditary problem, say for example, a person is not able to digest something because the enzyme producing gene is absent. So what if I insert the gene inside the person's body? So the problem is solved. That is gene therapy. Correction of a defect, genetic defect, involves delivery of a normal gene into the individual or the embryo to take over the function of or compensate for non-functional gene. So I am altering the defective gene or I am giving a new normal gene into the person who is having this hereditary disease. So we are going to talk about one such disease where gene therapy was first or initially tried. So that is called as the first clinical gene therapy, which was given to a 4-year-old girl who was suffering from a disease called as adenosine deaminase deficiency. So anything that ends in ASC is an enzyme. So when I say adenosine deaminase deficiency, it means that girl did not have this enzyme producing gene. The gene that codes for this enzyme is absent. So this enzyme is crucial for the immune system to function, right? So this disorder is caused due to the deletion of the gene for adenosine deaminase in some children cured by bone marrow transplantation. So many people, probably the occurrence of this is pretty high, okay? Okay, 4-year-old, okay, fine. Okay, so in some children, it can be cured by, hi Sudeshna, hello, yes, I'm very good, how are you doing? After a very long time you are here, so it gives me a lot of pressure to see you, so thank you so much for joining our session today. So please do like, share and subscribe, Sudeshna. Fine. So in some children, this problem can be cured by the bone marrow transplantation, because the bone marrow itself has a problem. So what if I change it and give a correct gene? But bone marrow transplant is not always successful. It's a very tedious and it's a very risky process. Others, it can be treated by enzyme replacement therapy. So I can replace the defective, or I can continuously give the enzyme so that this function happens, right? So that is enzyme replacement therapy, where I give functional ADA to the patient by injection. But both the approaches are not completely curable. So I have to go for an approach that actually makes sense. So in this case, I will insert the aDNA gene into the patient's genome. So that is the only curable way. It can be completely fine, and I don't have to repeat this process again and again. Right? Very good, Sudeshna. That's nice to hear from you. So this is how your so your ADA gene is involved in your purine metabolism. So we need your purines in the process of replication, DNA replication. So how is gene therapy done? So you take lymphocytes from the person who is suffering from this ADA deficiency. Curative means to cure. Okay. So curative means to cure. So you take the lymphocytes of the patient who is suffering from this ADA deficiency and you grow them in lab. Okay. At the same time, you take a retrovirus and you also isolate a functional ADA gene from another human, and you put this gene inside that retrovirus, because we also read in the previous chapter that for animals and humans we use retrovirus, right? So I take a retrovirus. So inside this I have this cDNA gene, ADA gene. So ADA gene, by recombinant DNA technology, I have it here and I'm putting it inside my retrovirus. So my retrovirus has my functional ADA gene and I'm putting this inside the lymphocytes that I have taken from this patient, right? So now see, now the functional gene is inside. So now I let it grow for some time. I insert it inside the baby. So this is one way I am using the recombinant technology. But the problem is lymphocytes are not immortal. They have limited lifespan. So once these lymphocytes die, then again I have to repeat this process. But this is how recombinant DNA technology can come into picture. But this is not permanent, right? So I have to do it in such a way that it becomes permanent. So in that case, what can I do? I have to treat it when the child is in the embryo stage, or I have to treat the stem cell of the child itself instead of this lymphocyte. But this is how your recombinant DNA technology is incorporated in the field of medicine. So today I spoke about how it is used in the field of insulin and how in the field of gene therapy. Right, so here we stop. How it is used in the field of diagnostics, I will continue on Friday. So until then, if you genuinely liked my teaching, so please do like, share and subscribe. Yes, I will teach evolution after this chapter is over. So we'll be done in 2 lectures. After that, I will start evolution. So thank you so much for joining my lecture today, guys. So stay tuned, take care until Friday. So this is me signing off. Thank you and bye.